Let's get this started. So I'm Kathy Yenley. I'm one of the directors for the Canadian Homebrewers Association. And yeah, welcome to this month's CHA speaker series. I'm super excited to have Bryan Heit on to talk about brewing with wild Canadian yeast. Just a little housekeeping before we start. If you can keep your microphones muted, just in case there are any background sounds that might come through the recording, because this is a recorded seminar. And we will have a bit of time at the end to do a Q&A. So kind of save your questions for then. You can ask it through the mic or do it through the chat, and one of us will bring it up if your microphone is not working. Bryan Heit is a scientist and microbiologist who has been homebrewing since 1996. While he loves homebrewing in general, Bryan specializes in vintage long-aged beers, mixed fermentation, and experimental brews with unique yeast, bacteria, and techniques. Bryan blogs extensively about brewing with his musings available at swaygenerousbrewing.com. I'm super excited to welcome Bryan Heit on here. Go ahead.
Great. Thank you, Kathy. I'm actually going to turn off my camera now that you've introduced me, because I'm out on a farm, so my internet's a little flaky here, but hopefully everything else runs fine. So like Kathy mentioned, my interests really lie in sort of wild brewing and wild yeast, mixed fermentation, those sorts of things. And so that's what I'm going to talk to you about tonight. So Kathy already mentioned this, but sort of by day, I'm a scientist, professor. I also am my wife's farm assistant, so I spend a lot of time doing that. And typically late at night is when I find time to brew, but I have been brewing for a while, so usually these things go all right. As Kathy also mentioned, I do run a blog, suigenerousbrewing.com, and I have a YouTube channel as well, which is YouTube/Sui Generis Brewing. So there's a fair amount of material that I've accumulated on those two sites over the past 5 or so years. And if there's anyone sort of known for, it's for doing sort of strange things. And I don't know how well these videos are coming across to you guys, but these are just 2 short clips from a video I put out last summer where I harvested honey from my own beehives, and I also isolated yeast from the same bees that made the honey and used that to make a mead. And so that's sort of an example of, I guess you would call it a wild mead rather than a wild beer, sort of brewed with pretty local ingredients. They're literally right out of my backyard. So, you know, I often get asked why I do these things. I think to a lot of people it seems a bit weird. You know, you can just go to the homebrew shop and get a nice clean yeast that's perfectly predictable. So why would you go and grab something out of the air? And I can think of a few reasons. One is, if you're ever interested in recreating any of this sort of classical Belgian sour styles or other related styles, they use spontaneous fermentation, where they collect yeast from the air and other places around their breweries. And so you can achieve a similar thing at home, but instead of, you know, taking a culture of their yeast, you can try what you have in your own yard or wherever it is you try to gather yeast from. It's also an opportunity to explore what may be unique about your area. So you could potentially combine not only wild yeast and bacteria that you collect, but also look at some local ingredients and see how those play together. You can also develop a unique house culture. And just as an example, my brewing club, the London Homebrewers Guild, actually has 2 wild yeasts as well as a lactobacillus that we have as a sort of guild-only yeast collection, and these were isolated by guild members and are sort of a unique thing that we have that most other brew clubs don't. And lastly, you might want to do this just because you can. You know, sometimes it's fun to try something new. So the other thing I kind of want to touch on at the beginning here is that Canada maybe has some fairly unique opportunities when it comes to brewing with wild yeasts as well as other brewing organisms. And that's because we have sort of a unique environment here not found really anywhere else in the world. And so if you look at a climatic map of Canada, we have what is probably one of the greatest diversity of climatic zones of any country in the world. Tropical rainforests around Vancouver and Victoria, all the way up course, to Arctic conditions in the north, various plains and taigas, and forest areas and all that as we go across the country. And the other thing that's fairly unique about Canada is that in almost all countries, climatic zones run from north to south. But we actually have not only that north-south set of climatic zones, but because Canada is so wide, we also get a climate gradation from east to west as well. And so that gives us a lot more diversity in climates than most other countries have. And so sort of by extension, there's probably a lot more things out there that we can brew with than most other countries would have access to. So just as an example of what we might be missing, because we don't do a lot of wild brewing in Canada, is just this one yeast here called Saccharomyces eubaryianus. This is actually a really important yeast because this is half of what makes up lager yeast. So lager yeast are a fusion between Saccharomyces eubaryianus and ale yeast, which is Saccharomyces cerevisiae. And so at some point in the distant past, these two things were in a fermenter, probably somewhere in Germany together, and they merged together to form a new strain of lager yeast. And until just a few years ago, we had no idea what the non-ale yeast portion of lager yeast was until Saccharomyces eubaryianus was discovered. And it was actually first discovered down in Patagonia. However, investigations since have found it in a number of other as in Mongolia. And basically this map, everything in green are regions of the world where this yeast might be found. And I would just point out that here in Canada, we have more green than probably any other country in the world. And this is just one yeast that has obviously very nice brewing properties to it. And who knows what else is out there. And so again, just to kind of emphasize this one last time, we are a country with incredible diversity in our climatic zones, in the different environments that we have access to. And so there's probably a lot of unique things across our country that we can brew with that we haven't really begun to explore. And so I think part of what I hope to maybe get people excited about today is the opportunity to perhaps start trying trying to see how some of the organisms in your local areas work when they're used in a brewery. So where would you look? And the answer is pretty much anywhere you can think. You can look in your backyard. You can look in your kitchen, although my experience in my house is I tend to find brewer's yeast and bread maker's yeast rather than wild yeast. You can look in parks, wilderness areas, gardens, farmlands, really anywhere you see plants growing is a place you could try to collect yeast from. And so sort of with that, I want to spend the next couple of slides just talking about exactly where to find them and sort of some general approaches to actually collecting these yeasts and beginning to use them. And then sort of the latter half is really going to be focusing more on specific methods and exactly how you would go about capturing and using these yeast. So if you consider sort of the average household, there's a lot of different places we can look for yeast. I mentioned already, you can look inside your home, but because we're all brewers, you're probably just going to find regular brewer's yeast in there. But if you start looking around outside, things like fruit, nuts, and the bark of trees tend to have wild yeast on them. As you can imagine, fruits that have a fair amount of sugar in them tend to have a fair amount of sugar-loving yeast on them, but bark is actually one of the natural environments of yeast as well. It's literally in the air. Every time the wind blows, yeast cells get picked up from wherever they're growing and they get blown through the air. Of course, gardens and orchards and vineyards and that tend to be to feed off the plants, and that's where they tend to be found. And actually, one of the best spots to get them, even if it's maybe a little bit weird, is from insects. Bees in particular, fruit flies, really any insect that sort of feeds on either the nectar of flowers or on the sugary juices inside of a plant will often have a lot of yeast within their intestinal tracts, and so they tend to be a good source for yeast, for finding yeast. And as an example, at the beginning, I used some bees. You can actually look in the soil as well as in leaf litter. And actually, we know for brewer's yeast, Saccharomyces cerevisiae, its original home before humans domesticated it was in leaf litter, and it literally survived by basically stealing sugars from the other organisms that were decomposing those leaves. Now, it is a little more dangerous to try and gather stuff out of soil or leaf litter because there are other organisms in there that have the potential to be harmful, but it is something that you can do as long as you set things up appropriately at the beginning to minimize some of those risks. So there's a lot of different places we can look. And as you can imagine, there's going to be very different organisms present in these different places. And it's not going to be single strains of yeast. It's going to be multiple strains of yeast, but it's also going to be other brewery organisms like lactobacillus and perhaps pediococcus. And so I don't know how many people here have done mixed fermentation, so I thought I'd talk briefly about what happens during these fermentations so you sort of know what to look for when. And so what I'm showing here is a graph that I sort of drew based on a couple of studies that looked at classic Belgian fermentations and what grows in these and in what order and in what timing. So what I'm plotting on the graph on the y-axis is just the relative concentration of the organism. So there's no numbers attached, it's just showing you where the peak of these cell numbers are during fermentation. And then time is along the bottom, starting at brew day. And one thing you might notice is things here are labeled in months. And if you haven't done mixed fermentations in the past, this might come as a bit of a shock. But most mixed fermentations take at least 12 months to complete, and 18 months or longer is not unusual. So on brew day, you gather your different wild organisms, and I'll talk about how you can do that in a few slides, and you add it to your wort. And the first thing that tends to grow during a wild fermentation are actually enteric bacteria and oxidative yeast. And these aren't things we want in the beer. So enteric bacteria includes things like E. coli and citrobacter. And so these are, one, potentially food poisoning organisms, but they also create a lot of unpleasant, unwanted off flavors. And then oxidative yeast, sometimes we want them. There are oxidative yeast in things like sherry floor, but they can cause a lot of like nail polish type flavors. So they're not something we really want much of. But the good news is they tend to disappear pretty quickly, usually within the first few days and certainly within the first week or so. And the reason why they disappear is because fermentative yeast grow. And when they start to grow, they of course start producing alcohol. They also acidify the fermenting beer. And you eventually get to a point where the alcohol and the acidity and the lack of oxygen kills off these unwanted organisms. So pretty quickly, those unwanted things should disappear, and you should now have a beer that has a lot of fermenting yeast. Now, some of these might be Saccharomyces cerevisiae, but there are a lot of other species of yeast that can engage in fermentation that might appear during this fermentation stage. Around the same time, we start to see lactobacillus appearing, and so this is why a lot of wild fermentations and mixed fermentations go sour, because lactobacillus is quite common in these beers. And lactobacillus will sort of grow and be present pretty much throughout the entire fermentation process. And as you can imagine, not only do these make the beer sour, but they're dropping the pH, and that further helps to get rid of these enteric bacteria and the oxidative yeast. And then at the later time points, two other organisms can appear. One of these is pediococcus. So this is another bacteria that acidifies the wort, quite often present in mixed fermentations. And you can also get Brettanomyces, which are a yeast that creates a lot of funky flavors, things like leather and tobacco, those sorts of things. But these don't always appear when you do a wild fermentation. In fact, Brettanomyces probably comes from the barrels that these beers are traditionally brewed in rather than from the air. And the pediococcus just sort of seems to be luck of the draw. Sometimes you get it, sometimes you don't. So when you're trying to capture and use wild yeast, there's a couple different approaches that you can use, and I'm going to go into these. But the one thing that's kind of worth noting is there's very different sort of time frames for when these processes can be performed. So some methods allow you to capture the yeast and sort of avoid the rest of it. And so those you typically are going to be looking around the 1 month time point or after. So relatively early in the process, you can start to actually sample those beers and start looking at what those yeasts are doing. But if you're doing a true mixed fermentation, if you're, say, trying to mimic a lambic or doing a golden sour, something like that, realistically speaking, you're going to be looking at that beer at 12 months of age or later. So I've mentioned already a number of times there's different options for capturing these wild yeasts, and there's lots of different ways we can go about doing it, and they all have their ups and they all have their downs. Some of them have a better chance of being successful, meaning you end up with a good beer in the end. Some of them have higher or lower levels of risk, and risk can be your beer going off, but risk can also be potentially isolating a harmful organism. And some of them have a better level of control over what you capture than other methods do. So I'm going to briefly use and sort of give you an idea of where they fall in those different categories in terms of risk, benefit, that sort of thing. So cool ships are a classic brewing method used by Belgian brewers in particular to capture yeast. You can also sample objects, flowers, fruits, insect, that sort of thing. You can also just sample the environment in general rather than targeting specific objects from an environment. And this isn't really a capture method, but once you have wild yeast captured, there are ways you can go about to purify them so you end up with single pure strains of yeast that you can then use in your brewery. So as I mentioned on the previous slide, each of these have different levels of risk, different chances of success, and different levels of control. And so if we look at risk, cool ships are sort of high risk in the sense that there is a bit of a chance, at least, of getting a beer that doesn't taste very good out of it. Not really any risk in terms of potentially isolating or potentially making a beer with a pathogen in it, but there is the risk of having a less good beer. When we look at sampling objects or sampling the environment, we have a much lower level of risk. We have a lot more control over what we're doing. And then when it comes to purified cultures, you really do need to plan things out properly and work ahead, or think ahead, sorry, because there is the possibility that you may isolate something dangerous. And now when it comes to success, cool ships have a bit of risk to them because of the chance of just making a bad beer. Purified cultures, if you do get into this, you'll find that you're going to screen a lot of yeast before you find one that actually tastes good. So the success rates on those tend to be relatively low. Whereas when it comes to sampling objects or sampling the environment, this almost always works out, usually because you do more than one sample, right? You sample a couple pieces of fruit, and at least one of those should give you a workable yeast. And then the level of control sort of is lowest with cool ships because you're literally just relying on things falling in the hopes that of getting some yeast, and then you get increasing level controls. And obviously, if you're purifying single strains, you now have total control over what's in your culture and how it behaves. So I've just mentioned sort of 4 different techniques we can use. So how do you actually go about doing these? So again, I'll start with cool ships. If you haven't or method of making sour mixed fermentation beers. So a lot of the lambic brewers in Belgium use this, as do some of the newer sour brewers here in North America, including companies like Jolly Pumpkin or Russian River. The idea here is you take your hot wort right after you're done brewing and you put it outside. And what you're relying on is the ambient cool air to cool that beer. And at the same time, you're relying on that ambient air to deliver the yeast and bacteria that will subsequently ferment that beer. So it's literally as simple as leaving a pot of hot wort outside, uncovered until it's chilled. My usual way of doing this is I'll brew in the evening. I brew on a propane burner out in the backyard, and I basically throw a barbecue grate on top of the pot when it's done so that raccoons can't get in, walk away, and I come back the next morning. So when it comes to sort of doing a cool ship properly, there's a few simple guidelines you want to follow. The first is you usually want relatively low IBUs in your wort, 3 to 5, typically speaking. And the reason for that is wild lactobacilli are not very hop tolerant. And so if you want sourness to develop in the beer, you have to keep your IBUs low. You can use aged hops as well, and you can get away with a larger amount of aged hops. But again, it's not like if you buy a mixed culture like Rosalaire from Yeast, where those are more hop tolerant lactobacilli. You still need to keep the IBUs in check even if you're using aged hops. Your starting gravity can cover a pretty broad range, anywhere from 1035 to 1065 or even higher. I've done some of these as high as 1082 or 1083. I don't remember exactly. But when you get to those higher gravities, it is a little bit more challenging to get these things to ferment out the way you'd like. But if you stick to that 1035 to 1065 range, it's a pretty reliable range. I mentioned this in the previous slide that I put a heavy barbecue grate on top of my pot when I do this. And that's just because we have raccoons running around and I don't want one to get in. But you do want something that's got a relatively coarse grating on it, because one of the major things that might carry yeast into your cooling beer are actually insects. And so you do want them to be able to get in. Now, historically, it's always been said that this method works best when nighttime temperatures are getting down near freezing. My experience has been not quite so strict as that. I found that so long as the nighttime temperatures are getting down to ale fermentation temperatures, so 16 to 18 Celsius, it seems to work fine. I've never had any issues with not having, 1 or 2 degrees Celsius evenings. It seems to be fine if it gets down to 16 or 18. And I've actually had a few where the beer froze overnight because it went below freezing, and it still started fermenting within a couple of days. So you can even even do this when the when the weather is getting on the colder side. And just a few other sort of hints and tricks if you're going to do some cool ship brewing. Probably the easiest way to do this is to cool overnight. It gives you plenty of time for the beer to cool, plenty of time for it to gather yeast, but you can do it during the day as well. Just keep in mind that you're going from boiling to fermentation temperatures. It's going to take a while, even on a relatively cold day, and you want to also make sure there's enough time for it to become inoculated properly. Now, once you're ready to transfer it into your fermenter, you're going to want to transfer it into a carboy, and you're going to want to cap that with an airlock. Once the beer has been inoculated, it's really important you limit the amount of oxygen present. The reason for that is, one, some of those oxidative yeast can grow and cause off flavors, but there's also a risk of mold growing as well. And if you get significant amounts of mold in a fermenter, you unfortunately need to dump that beer because mold can be quite hazardous. And I just want to point out, in this pot here, you might notice there's this funky looking mass of stuff. That's not mold. That's actually what your hot break is going to look like if you cool ship. So it tends to form these really large rafts of material. But they're kind of nice because it actually makes it very easy to then pull off clear wort. And you can kind of see next to this large raft here just how clear that wort's become in this overnight chill. Once you've got it in a carboy, you want to keep it at ale temperature, so 18 to 22 Celsius, and you want to let it sit at least 2 weeks to allow fermentation to occur. Sometimes fermentation can take a while to get going, and you'll know it's fermenting because you'll have bubbles in the airlock. You'll see the wort go cloudy because of the yeast. You might start to see yeast settling out. If at the 2-week mark you don't see visible signs of fermentation and it's just sitting there, you might want to then consider pitching an ale yeast, in particular a Belgian ale yeast or a Saison yeast, to help kickstart things. And you should still get some wild character out of it, but if in 2 weeks it hasn't started fermenting, that's a sign it's going to need some help. And so you're going to want to add some additional yeast. And then you basically forget about it and just top up the airlock every time it goes dry. 12, 18 months later, you'll have yourself the beginnings of a sour beer. Now, one thing I will say is don't expect what you get when you buy a Lambic or Flanders Red or something like that from the store, because those beers are actually made by doing multiple batches of beer and then blending them together. Unless you're going to have a large sour program where you have a lot of different carboys of sour beer on the go, that's not really an option. And so the beers that these tend to produce, if you're just doing a single carboy's worth, tend to be a little one-dimensional. Some of them will get really sour, but not with a lot of funk. Some of them will develop a lot of funk, but maybe lack a little bit on the sourness side. And so usually once you get to the point where it's sort of done, you do need to do a little bit of extra to it. So that might be adding some fruit or adding some spice or blending it with another beer to bring out a bit more balance. And so, for example, saisons are great beers to blend with these types of cool shipped ales. But at the end of the day, you can end up with a pretty tasty beer. Just keep in mind that once fermentation might not be done, sort of fine-tuning that beer. So that's cool ships. Sort of the next way of sampling or collecting wild yeast is what I call environmental sampling. This is a fairly simple and straightforward process. So typically what I'll do is I'll prepare in a half-liter jar to a 1-liter jar some wort, and I will pressure can it so that it's sterile, and I just keep these on the shelves. And when I want to do a sample, I'll open it up and I'll put a mesh on like this to keep bugs out if the goal is to keep bugs out, or I'll put a more coarse mesh on it to keep squirrels out if I do want to get some bugs in there. And then you just put it somewhere that you think you might have some yeast kicking about, and you let it sit there for about 12 hours. So I've placed this jar in my wife's old garden at her old house and collected yeast that way. And basically, while it's sitting there, air currents will carry yeast in. If you do have a mesh large enough, bugs can get in, the bugs will bring yeast in. And you can then transfer that into a fermenter with an airlock, and you let it run at least a month, maybe a little bit longer. And that's enough time so that those enteric bacteria, as well as non-fermentative or poorly fermentative yeast, die off, at which point you now actually have a mixture of brewer's yeast, or at least beer-friendly yeast, as well as probably some lactobacilli, things like that. And you can now start doing small-scale or full-scale ferments to see what it tastes like and to see if it's worth carrying on further with. And so I actually really like this method. I don't do too many cool ships anymore. This takes a lot of the risk out, because you can imagine you set up 5 or 6 of these jars around your yard, giving you 5 or 6 different cultures. You then do 5 or 6 little mini ferments, and you see what they taste like, and you can then move forward with your full size batch with a culture that doesn't taste horrible. It may not be fully developed because, as I mentioned before, these beers often take 12 or more months to develop fully, but you'll at least know up front that your culture has some positive aspect to it. You can also capture from objects. Fruits are excellent, flowers are excellent. Here I have a wild grape and a hop cone, tomatoes, raspberries, fruit. You can do leaves, you can do bark, any object you think that might have yeast on it. You just put them in a tube. These are just sterile centrifuge tubes that I buy off eBay. You can, again, just use mason jars that are clean. It doesn't have to be a sciencey tube like I have here. And you pour some wort on it. If you're looking to make a wine or mead, you may actually want to pour a juice onto it instead, so that it's more similar to the product or to the beverage you want to ferment. And again, after 6 to 12 hours, you actually want to remove the object, because I've found if you leave them in there, they tend to be sites where mold will start to grow, and you don't want mold in these things. And then you transfer to a fermenter, or if it's in a tube like this, you can just plug an airlock right on top. And just like before, you let this go for a month, and at the end of that month, or longer, you can then run little mini ferments and decide on which particular culture you then want to use in your beer or mead or cider, whatever it is you're brewing. So that's 3 different methods of getting wild yeast, or getting a wild culture. And then the next thing you want to do is assessing what you have. And so if you're cool shipping, this is the assessment you're doing when the beer is done. If you gathered from objects or if you've collected from the environment, this is what you would want to do before, either before you start the test ferments itself, or it's what you would test or do with the test ferments. And it's really important you're careful when you're doing this, because there is a very small probability, but it's not a non-zero probability, that you may accidentally collect something potentially hazardous. So it's really critical you assess with all of your senses other than taste before you start tasting something. The other thing I would recommend, especially if you're doing a test ferment to see what your culture looks like, is trying to use a neutral base beer. So I like to use just a 50-50 Pilsner wheat malt mix, about 20 IBUs of whatever hop I have lying around, 1045, 1050 gravity, and that's what I tend to use for my sort of standard test beer. You don't want anything too crazy on the malts or the hops, because you may not then sort of be able to tell what's going on with the yeast you collected. Obviously, once you have your yeast and you've decided that they're worth fermenting with, you of course can design a beer to match the particular characteristics of the yeast. But when you're testing them initially, you want to make sure you're using a neutral beer so that you're really just assessing the yeast on their own. So the first thing you want to do is just look at it. Does it look right? It should look like beer. It might be cloudy, it might be clear, but it should look like beer. It may have a pellicle on the top, which is a little floating raft of yeast and bacteria together. If that pellicle is white or gray or beige, that's fine. That's totally normal. It's what you'll often see on mixed fermentations in a carboy. You may even see little slimy fingers kind of hanging down from that pellicle. And that's actually fine as well. Wild beers tend to go through a stage that we call being sick, and that's where some of the bacteria in it that make it sour also make that sort of mucousy-like material, and eventually the yeast just break that down and convert it back into beer. So if you see slimy fingers in it, it's probably okay, although you might want to be a little more cautious moving forward. If you see mold, if it's gelled or gone solid in some way, shape, or form, or if the pellicle or the sediment in it is colored, anything other than sort of a white to beige tone, dump it, because nothing that makes beer is colored, whereas a lot of things that can make you sick have color to them, especially things that are red or green or black, you really want to avoid. So if it looks good, the next step is to smell it. And again, you're assessing beer, so you should know what the smells like. It may smell just like a regular everyday beer. I've had some that smell no different from a Budweiser or a Molson. You wouldn't know it was wild yeast. But these are wild yeast, so sometimes they do have exciting characteristics. They can be fruity, they can be spicy, they can have funky notes, which can range from earthy to leathery, tobacco, those kind of things. Those are all fine. That's what we would expect to be in there. Now, if it smells of anything bad, and these things can smell really bad, you want to dump it. Things like the smells of vomit or feces or urine or body odor are signs that those enteric bacteria took over and the yeast did not. Corpses or rotting meat, that's another thing. You don't want to be brewing with that. Ironically, the things that make these aromas are actually not harmful to us, but do you really want a beer that smells like a corpse? Really, if it doesn't smell good, you don't want to work with it. The third thing you want to assess is if it attenuated, because this is really an important part of getting rid of anything potentially dangerous within that sample. So if you have more than 60% attenuation, which would be going from 1040 to 1015, or if whatever level of attenuation you've had has given you more than 4% alcohol, you should be fine to move on. If you're in the 40 to 60% attenuation range, or you're above 2% but under 4% alcohol, I wouldn't use that sample as is, but you could add, a Belgian yeast strain or an ale yeast to it, let it ferment out, and can then use that blend moving forward. You may still have some interesting flavors in there, but the yeast that are in there aren't quite potent enough for what we're trying to achieve. Now, if it's less than 40% attenuation or less than 2% alcohol, you want to dump it. You really do need more than 2% alcohol to get rid of those potentially dangerous organisms from your wild capture. Now, the fourth thing is optional. but if you have a pH meter, it can be a useful thing to look at. And you just want to look to see if the pH is below 4.5. So if it's below 4.5, again, this will help to suppress anything dangerous. You're fine to move on. If you don't know, but all the other tests you did, the attenuation, the aroma, the appearance, were fine, you're good to move on. But if you do measure the pH and it's not below 4.5, you really don't want to continue on because, again, you may have harmful organisms present. And then it's time to taste it. And when you taste, obviously start with a small sample. Don't pour yourself a pint, and assess carefully. What's the aroma? What's the flavor? What's the mouthfeel like? And what you're looking for are all the classic aromas and flavors and mouthfeels you would have with beer. If you're familiar with sour beer or barrel-aged beer, a lot of these more unusual flavors and aromas will be familiar to you. If you're kind of new to the area, they might come as a bit of a surprise, but they are quite pleasant if they're in balance with the other flavors. And if it tastes good, smells good, and looks good, you should be fine to move forward and either brew a full beer with it, or if this is from a cool ship, you should be fine to tweak it in whatever way you want to tweak it, and then keg or bottle that. Excuse me. So everything I've told you up to this point is really sort of the very simple entry-level way of doing these captures. But there's a few little things we can do that can really help improve our chances and improve our safety. So I would really recommend engaging in some of these practices. So this table here, I know there's a lot going on, but it's basically pointing out that organisms like enteric bacteria, so these are those ones that create a lot of off flavors and also can be potential food poisoning organisms, are suppressed at pHs below 4.5, and they're suppressed by alcohol. Molds and oxidative yeasts are suppressed by low pH and suppressed when oxygen is absent. Often our wild captures, things like lactic acid bacteria, are suppressed by IBUs, typically 10 or more will prevent them from growing. And our fermentive yeasts are basically tolerant to most of the conditions we would see in beer. And so because we know where the things that we don't want sort of struggle, and we know what conditions the things we do want thrive in, we can actually tweak our capture, the way we set up our wort before we do our capture, to help out the organisms we want and to suppress the organisms we want to avoid. So probably the most basic thing you can do is pre-acidify your wort. So whenever I do a wild capture, whether I'm sampling from fruit or just putting a jar out in the yard to see what falls into it, I'll use 1040 gravity wort that I pre-pH to pH 4.5 using lactic or phosphoric acid. And then I just move forward in exactly the way I told you earlier in the talk. I put it outside for 12 hours, or I soak the fruit or whatever in it for 12 hours and then put it into a flask with an airlock. You can also do this if you're cool shipping beers. You can pre-pH the beer to 4.5 with lactic acid or phosphoric acid while it's boiling. And then when you do the cool shipping part of the process, it's now protected from those enteric bacteria, and there's a reduced risk of mold as well. And in fact, in Belgium, some brewers are starting to do this just as a way to reduce the risks that come with those enteric bacteria, both in terms of the off flavors they can create, but also in terms of the risk they may present to their customers. You can tweak your wort to actually help select for yeast. Let's say you don't want those souring bacteria and you really are just after the yeast. Well, again, you can start off with a 1040 gravity wort at a pH of 4.5, but you add to that 15 or more IBUs of hops, and this will suppress most wild lactobacilli and should give you a purer yeast culture than you would get if you didn't have hops present. Again, once you've done the capture, you still want to make sure that you're limiting oxygen exposure by using an airlock. You can also set things up to try and get more alcohol tolerant yeast. And this is particularly useful if you're trying to make, say, a wild mead or a wild wine. And so the way that I do this is I'll make a wort that's at a gravity of about 1050 with a pH of about 4.5. If I don't want lactobacilli in it, again, I'll hop it. And then what I'll do is once that wort has been boiled and cooled, I'll add to it 15 mils of 40% vodka for every 100 mils of wort that I'm using in my capture. So it's a 15% solution of wort. And what this does is it lowers the starting gravity of the wort to 1040, but you start off with a 6% alcohol by volume solution. And so by the time the yeast is done fermenting this, it's going to be around 9 or 10% alcohol. And obviously only yeasts that are fairly tolerant to high alcohol levels will make it through that. And so it is a good way to sort of pre-select ahead of time for alcohol tolerant yeast. And likewise, you know, if it doesn't attenuate fully, that those yeasts probably aren't going to work very well if you're trying to make a mead or a wine where your alcohol content is going to be, say, 8 to 10% or 12%. And once again, once it's inoculated, you want to limit oxygen exposure with an airlock. So once you have a call, had a few that I've not kept in the past that I regret not keeping. And there's different methods of doing this. So one method is freezing. This allows you to maintain both pure cultures if you've purified a yeast, or mixed cultures if you have, you know, one of these wild captures. The advantage to freezing is it avoids drifts. So wild cultures tend to drift over time, both because the relative portion of the different yeasts and bacteria in that culture tend to change over time. But also it avoids any sort of genetic drift that might occur as the yeast and bacteria evolve to survive better in beer. And I know freezing yeast seems like it's probably fairly complicated, but it can actually be done with pretty simple tools and pretty inexpensive equipment. And so on my YouTube page, I do actually have a dedicated video on how to freeze yeast. So if that's something of interest to you, I encourage you to check out that video. And it doesn't have to be wild yeast. This is also a way you can just preserve your day-to-day brewer's yeast if you want to, say, hold on to one of your favorite yeasts for future batches. A lot of brewers will lean towards agar slants or agar plates for trying to store cultures. These are actually best for pure cultures. It's very hard to maintain a mixed culture where you might have a dozen different types of yeast and a couple different kinds of bacteria on a plate or a slant. Oftentimes when you sort of recover your culture from that, you'll have lost some of those strains. They do have the advantage that they limit drift. They're not as good at it as freezing, but they do limit the amount of change in that culture. And even though the impression is that this is an easy method, I would argue it's actually more difficult than freezing. And it certainly requires a little bit more in terms of equipment. And then by far the simplest thing you can do is just keep repitching that culture. You can, you know, brew another beer and dump it on the yeast cake, or you can open up a bottle of the beer and dump the dregs into a starter and then dump that into a beer. And that's another way of maintaining a mixed culture. You can also use it to maintain a pure culture. The downside to this, though, is you really drive the drift or the change in that culture when you do that. Basically, every time you pitch it, the balance of yeast, the different yeast in there and the different bacteria in there will change, and that can then change the way the beer tastes. But it is really easy. It's about as easy as it can be. So that's sort of the basics, how you would identify a site where you might want to try to find some wild yeast, way to collect those wild yeast, ways to sort of tweak the method. And so I just kind of want to finish up my last little bit of my talk by talking about a little bit more advanced methods that we can use to really kind of give us the best chance of success and to really try and enrich for yeast that are going to thrive in a beer. And so this is using a method called boot camping, which is a method from South Yeast Labs. And the idea behind boot camping is you're going to force whatever's in that mixed culture, the yeast and the bacteria, to ferment wort under increasingly difficult conditions. So it's harder and harder and harder at every step in the process for the yeast to ferment and to survive, or if there's bacteria in there for them to ferment and survive as well. And so essentially, you might start off with 100 different yeast in that culture, but by the end, there might only be 2 or 3 different kinds of yeast in that culture because they've outcompeted all the other yeasts by being the ones that can sort of survive the boot camp. So in this video here, this is me setting up sort of a boot camp culture. In this case, I'm adding vodka both to myself and to the starter. And the point of the vodka is, like I mentioned a couple slides ago, is we're trying to push up the alcohol level in the starter to make sure that the yeast coming out of it are quite alcohol tolerant. So David Thornton's method's fairly straightforward, and his method is meant to be sort of a rapid way of taking something like a fruit or a leaf and getting the yeast out of it and selecting the most fermentable, beer-friendly yeast possible, sort of as quickly as possible. And so the way that he does it is you take your fruit or your bug or whatever it is you're trying to get the yeast from, and you add it to a half cup of low-gravity wort, something around 1024, and you want to put it in a flask that has a foil cap, because early on you actually want a lot of oxygen in here to help those yeast get going. As soon as you have visible fermentation, though, you replace that foil cap with an airlock in order to now start reducing the oxygen levels and to start putting some pressure on the yeast. Once fermentation is complete and the yeast have settled, you decant, which means you're pouring the liquid portion off but leaving the sediment behind. And you're now going to add a half cup of higher gravity wort, something around 1040, put the airlock on, and again let it ferment to completion. You then decant again, put another half cup of wort, this time really high gravity wort, 1085 or thereabouts, and you cap with an airlock and let it ferment. And once the fermentation is complete, you collect the yeast, and you should have highly fermentive yeast. So David Thornton recommends you actually grab the yeast that's still in suspension at the end of that fermentation period, because in theory, that's the stuff that should be sort of the most robust and the most able to survive in these higher gravities. And from there, you can either purify further if you're interested in trying to get a pure strain out, or you can start testing it in some ferments to see what it tastes like. So I have a slightly different method from this, but it's sort of the same thing, sort of the same idea. The difference is I often am not selecting for higher gravity, I'm selecting for fermentation speed. And so I usually start off actually with a month-old wild capture, something that I've captured from fruit or captured from the environment, whatever, like I showed you earlier in the talk. And the reason why I like to start with that 1 month-old culture is I can do my initial assessment on it and see if there's actually something in there worth purifying. The one downside with David Thornton's method is you're kind of going in blind and just hoping there's something good there. But if you have a little mini ferment that you've done and you know that it tastes okay and it smells nice, there's something in there you might want, and now you can go after it. So what I'll do is I'll take about a tablespoon of that wild ferment, or if I already have it down to a yeast sediment, about a half teaspoon of the yeast slurry, and I'll add it to a half cup to a cup of wort, somewhere in the range of 1040 to 1060, and I'll immediately cap it with an airlock. I let it ferment for a day, and then I take out a half teaspoon, so a very small amount of the fermenting wort, and I add it to a fresh flask containing 125 mils of wort at roughly the same gravity, cap that with an airlock. I might repeat that an additional time or two, and then I'll let it ferment to completion and collect the yeast. And what I've done here is I've really kind of selected for yeast that are fast fermenters, rather than what David Thornton does, which is select for yeast that are able to ferment to higher gravity. Of course, you could always ramp up the gravity as you're doing these different steps if you were also interested in trying to get a higher gravity tolerant yeast. So the thing that you end up with after boot camping is of alcohol tolerant yeast. There may or may not be bacteria left at the end of that process. If you use hopped wort, there probably won't be much, if any, bacteria left. So it's not necessarily a good approach if you want a sour culture, but it is a way of getting a good yeast culture out. Now, you're not going to have a pure culture in the sense that it's not going to be a pure strain like US05 or something like that. There is going to be more than one strain and probably more than one species of yeast in there, but it is going to be biased towards yeast that are tolerant under the conditions that you've set up. So if you've set up high gravity conditions, they're going to be yeast that are tolerant towards high gravity beers. If you've done my method where you're sort of forcing them to ferment quickly, you should have fast fermenting yeast. If you combine those two methods, you should have fast fermenting yeast that like high gravity. So that can serve two useful purposes. One is you're going to hopefully have an improved mixed culture that's going to be more vigorous. It's going to produce beer more quickly, quicker maturing beer. So it's a way of getting a culture that should give you a wild beer in less than 18 months. And I've actually had a few cultures I've prepared in this way that basically ferment on the same sort of timelines that you would ferment an ale on. So 2 weeks, 3 weeks later, or 2 weeks, 3 weeks after brewing, you can actually cake these beers. And if you're interested in taking the next step in the process, which is actually purifying single strains of yeast so that you literally have a pure strain, you know, your house strain equivalent to US-05, but it's your house strain, this is the good spot to start because you're going to set yourself up for success when it comes to purifying those strains. Now, I'm not going to talk today about purifying single strains. That's like a whole 2-hour topic in and of itself. But I do have on my YouTube page 2 video series that will take you through that whole process if it's something you want to try. So the first one is the Hunting Wild Yeast video series, which if I recall is a 3-part series. And it basically takes you through the capture portion, which is what I talked about today. The isolating individual strains of yeast from your capture. So here on this plate, you can see I've got a grid of about a dozen different strains that I've purified. And you can't see it here, but the final one talks about characterizing those yeasts and determining if they're good for brewing. The other video series I have that will be useful if you want to try of yeast is my Home Yeast Lab Made Easy series. This is a video series that's intended to teach you the basic microbiological techniques that you need to purify yeast, as well as to do other things with them, like freeze them or store them on slants. And it's literally a video series that is going to take you right from the very beginning. So you don't have to have any previous lab experience, any previous microbiology experience at all. It'll take you from the very first steps right through to the end. It also shows you in a couple of the videos how to make some homemade equipment in order to save some money. So with that, I think I'm done. So I don't know how to get my video feedback, but if there's any questions, I'm happy to take them. There we are.
Can I ask some dumb questions?
There are no dumb questions.
Great. Usually someone says it right before a dumb question. But so 2 things. I love, first of all, thanks a lot for your series and your videos because they're fantastic. So I'm not a biologist. My son with me is, he's like doing his PhD, but I'm not. So a couple questions. One is that for all kinds of yeast, and I'm not sure because you have videos both on slants and freezing, what would you say is actually like the best, like if you have to pick one method, because I'm just at the plating stage as a rank amateur, what would you pick?
So it really depends on how much you plan on using it. The downside to slants is every 3 or so months you actually have to make a new slant and then take the yeast from the old slant, put it on the new slant, and kind of let it grow, and then you can cap it and put it back in the fridge. So it's a lot of work for a yeast if you're only going to use it, you know, once a year or once every 2 years. But if it's a house culture that you're going to be using frequently, then it's easier because with the frozen yeast, it's a little more involved to get the yeast out. So it depends on your usage. And the other thing too is it also depends on how many yeast you want to keep. So when I first started, I started with slants and I maxed out at about 10 yeasts. After that, it was just too much to keep track of how old they were and when they needed to be recultured and all those different things. Since moving to freezing, I have somewhere in the neighborhood of 400 or 500 strains of yeast, and it's no effort at all because they're in the freezer. They don't do anything, they just sit there. So you know, it depends on how much you want to use those cultures, how many cultures you expect to have. You know, with the freezer, you have to set a little bit more up in advance because most home freezers have a defrost cycle, and that's actually really harmful if you're trying to freeze yeast. So you almost have to have them in a cooler in the freezer so that, you know, they're protected from that temperature fluctuation. So it depends on what exactly you want to do and how much effort you want to put into it and how many strains you want. So you kind of got to think your way through that and pick appropriately.
Okay, then the second one is a dumb question. My son who's sitting beside me is going to cringe, but if you have a slant or something, so you have like you've not autoclave, but like used a pressure cooker to do mineral oil, whatever, something you put on top of it.
Mm-hmm.
How do you get the yeast? Do you dump the oil, add
Nope.
more in, but how do you get the yeast out of it?
So you literally, sanitize your wire loop in a flame, let it cool, and then just pick the yeast right up through the mineral oil, and then right into your small starter.
Awesome. Okay, thanks a lot.
No problem. Anyone else?
Yeah, so when you're doing frozen cultures, are you putting them under glycerol, or are you putting them in glycerol before freezing them?
Yes. Yeah, so if you don't do that, it's a really efficient way of killing your yeast. So again, I have a whole video on it, but the short version is you want to take wort, not too high gravity, 1030, 1040, and you want to add 40% by volume glycerol to that. And then when you freeze your yeast, usually I start with a mason jar of yeast. You want to add an equal volume. So you got your mason jar of yeast, you pour off as much of the old wort as you can, and you then want to put in as close to an equal volume of that wort glycerol mix as there is yeast. So you want like a 50-50 mix. So you should have about 20% glycerol once everything's mixed together, and then I pour it in the tubes and put it in the freezer.
And are you sterile filtering or autoclaving that glycerol, or are you
Yep.
just
So you can autoclave the glycerol or pressure cook the glycerol. You don't need an autoclave. You can also, honestly, just standard canning boiling is usually enough. You don't always have to go to the extent of pressure cooking. And you can either, it depends on how much you want to have sort of set up ahead of time. You can actually pre-mix the glycerol and the wort in a jar of some sort, pressure can that, and that'll then be stable for months. And as long as you're careful with it, you can do multiple rounds of you can use it to make multiple batches of frozen yeast just by carefully pouring it off using a Bunsen burner to try and protect it from contamination. Or you can just make it up when you need it. But you can even just autoclave or pressure cook the glycerol itself in its pure form.
Okay, and the final part. So when you're done, when you go to pull something from your freezer, are you then streaking that out onto a plate before using it, or are you just taking that culture and inoculating it?
I'm too lazy to streak it out on the plate. Usually, so I freeze yeast in two different ways. So I have like a yeast bank, which I mentioned before. I have like 400 strains of yeast in it, and they're in these tiny little tubes. They hold about 1.5 mils. And so there, what I'll do is I'll take a wire bacteriological loop. I'll use a Bunsen burner to sterilize that, and then once it cools, scoop a little bit of it out, put it into a small tube of wort, and let that grow up, and that then becomes the basis for a starter. But the other thing I do is there's a couple of yeast strains that I just use quite a bit, like W3470, and so for that, what I'll often do is I'll take a starter and I will dilute it, suspend it in the glycerol wort mix, and then I'll actually freeze it in old white lab tubes is actually what I like to use for that. So that's closer to a pitchable amount, and essentially what I can do is I can thaw that whole tube, dump it into a 1 liter starter, and within a day or two I have enough yeast for a brew day. So it depends on how you want to manage it.
Perfect, thank you.
Yep. Anyone else?
So you had mentioned while we were waiting for this to start that you made a wild mead. How did that go?
It's pretty good. It's a little tart, but in a good way. One thing with wild mead or wild beer is they tend to take a long time to mature. So I've made that in, I think, September of 2018. So it's really only just coming into its prime now. You know, it's at about, you know, what, 18 months, I guess, 16 months. So it's really starting to get good now. You know, they tend to be a little rough around the edges before then, but it's quite nice.
Do you do the yeast nutrient routine with meads?
Yep. So I do Tonza or Tosna, whatever they call that, or something close. I'm not sure I'm exactly doing it the way the full way that they spell it out. And certainly, I mean, not just with meads, but ciders and even beers. One thing to keep in mind with wild yeast is they tend to be relatively high nutrient demand yeast. So if you're doing like a Tosna style yeast dish or nutrient addition, make sure you're using the calculation with with the setting set for a high nutrient requiring yeast, not a low nutrient requiring yeast. Otherwise, they will really struggle, especially in something like mead where there's really not much there other than sugar. But even in beers, if you're trying to push the alcohol content up above about 8%, I'd recommend adding some yeast nutrient to it because they will struggle more than your sort of standard brewer's yeast.
One more quick question, because we have a couple hives out back, and and I noticed that you've done your bee capture. Did you like grab bees that have just died? Did you grab a few live bees and put them in?
They were alive when I grabbed them. I just went out with a jar and I opened up a hive and put the jar over top. And, you know, they naturally like to climb up, right? So about 5 of them climbed into the jar, and I stuck them in the fridge for a few hours to humanely euthanize them, I guess you would say. And then, yeah, you took the yeast from there. You wouldn't want to take one that had died because it would probably already have started to mold. And, you know, you want yeast, not mold. So
Gotcha. We're just laughing here, the two of us, because we always feel guilty because, you know, you get like one flies on your window in the winter and you're like thinking, oh, man.
yeah, I know they're my babies, but I still killed 5 of them anyways. I mean, there was like 40,000 other ones flying around. They're fine, but
Any other questions? Cool. Well, we might wrap it up now. Oh, was that someone with a question?
no, I think that was my phone with an email.
All right, then. Well, Bryan, thank you very much for coming on and chatting about this. I think this is really great information and super accessible. Like, I'm really wanting to try to cultivate some wild yeast instead of just dumping things in a carboy and hoping for the best.
All right. Well, thank you so much for having me. And, you know, if anyone has questions, feel free to email me. I know my webpage is a horrible name, but if you go on there, there is my contact email on there, and just send me an email if you have any other questions. I'm always happy to answer. Or if there's a question to a specific video on YouTube, I'm pretty responsive at replying to comments on there as well.
Awesome. Well, thank you very much, and good night, everybody.
All right. Good night, everyone. Thank you for joining me.
Thank you very much.